Morning, everyone. Firstly, I would like to thank you for this opportunity, and it's my pleasure to introduce myself. My name is Krishna Kumar, PhD Scholar of Agriculture and Entomology from the University of Agriculture Sciences, Bangalore. I'm here today to present on the topic volatile chemical signals underlying the host plant preference of Earias vittella in Abelmascus species. Certainly, insect cannot think, but they can react. According to this saying, when herbivorous and carnivorous insects are looking for food, mates, and ovipositional site, they use chemicals as a communication signals to locate their sources. In the light of foregoing, the objective of this study was to understand the volatile chemical signals underlying the insect-plant relationship. Here, the Earias vittella, commonly called as okra, shoot and fruit borer, used as a test insect on different host plants of Abelmascus species. Because the damage of this insect were recorded the range of 12 to 90% in the okra ecosystem in different parts of India. The methodology used here as the host plants for this preference study, 10 different species of Abelmascus were used, where 3 of them are wild species, 6 are genotypes, and the name is Arca anamica, is a cultivated variety. In case of ovipositional preference, there are 2 kind of choice assay were employed. The first one is choice assay. In choice assay, 10 alternative host plants were exposed to the gravid female moth to lay the eggs. In case of no choice assay, each of the 6 chosen host plants were exposed to the gravid female moth, one at the same time. And the 72 hours of exposure of this moth to the host plants, the mean number of eggs laid on the host plant were recorded. For the identification of host plant volatiles, the fresh host plants were used for the volatile collection by employing air entertainment system. Further, these volatiles examined for behavioral assay by using white tube olfactometer. Here is the olfactometer, one arm is treated and the another one is controlled. The gravid female moths released into the white tube olfactometer and the behavioral response were recorded. Further, the most influencing host plant volatiles were screened by using olfactometer, and this screened host plant volatile used for the antenographic detection to identify the responsive volatile chemical compound for the female moth attraction. So here the results comes. The ovipositional preference results of no choice assay were plotted here as x-axis shows different host plants and the y-axis shows number of eggs laid. The data were analyzed through the one-way ANOVA, and it shows that the Arca anamica is a cultivated variety of okra or lady's finger, harboring the more number of eggs significantly. The trend follows same in the choice assay too, by having the huge number of variation, by having the more number of eggs on Arca anamica, a cultivated variety of lady's finger. Here, the next result for the white tube olfactometer bioassay projected. The graph here shows in the x-axis, there are different host plants were plotted, and the negative and positive behavioral response of gravid female were plotted in the y-axis. Chi-square tests were followed for this data, and it was highlighted by 4 genotypes were found to be a highly attractant, and the 3 wild species were found to be shows the negative behavioral response of gravid female moths. By comparing both ovipositional preference bioassays and the behavioral bioassay employed by white tube olfactometer test, the host plant of Arca anamica, it's a cultivated variety, shows more attraction towards the gravid female moth. Further, this host plant volatile used for antenographic detection. The antenographic detection used here as by employing gas chromatography coupled with electro-antenographic detection. So here the result of AGCAD shows obvious and obvious antennal response of female moth to the correspondent volatile compound. There are 17 major compounds were found to be attraction through this result of GCAD. In that, 7 compounds were found to be synthetically available. Further, the evaluation of these synthetic chemicals individually or in combination of combination under the various concentration should be analyzed in the field condition for the potential attraction. And that brings me to the end of my presentation here. Let me give you a summary of what I have been presented over the last few minutes. Host plant preference of gravid female moth of Earias vittella analyzed and a different experiment, namely ovipositional preference assay and the behavior bioassay, shows a significant significant attractive volatile compound. These identify specific volatile compounds individually or in the blends that can elicit positive behavioral response in the female moth of Earias vittella. Such compounds can be utilized for developing the bait traps in the future. And it also can serve as a potential component of the sustainable management in concern of environmental safety. So here I would like to acknowledge Indian Council of Agriculture Research for providing me senior research fellowship, Dr. P.D. Kamala Jayanti, my major advisor, and Indian Institute of Horticulture Research for providing the research facility. And finally, I thank University of Agriculture Sciences, Bangalore, and Dalhousie University for providing this wonderful opportunity for present my topic here.